Welcome back to the deep dive. You know, if there's one medication that's pretty much guaranteed to be in every medicine cabinet, well, it's acetaminophen.
Absolutely. Tylenol here in the U.S., Paracetamol in many other places. It's just ubiquitous.
It really is. It's a go-to for fever, for pain, and especially for pregnant individuals, it's always been seen as, the safe option, one of the very few drugs considered okay.
That perceived safety, going back decades, is exactly why this conversation is so important and, frankly, a bit unsettling.
Yeah, it really creates a dilemma, doesn't it? Because estimates suggest something like 65% of pregnant women in developed nations use it.
It's almost unavoidable in some situations.
Uh,
But now we have this growing body of evidence, and we're talking top-tier research here, systematic reviews, meta-analyses.
right, the studies that look at all the other studies?
Exactly, and they're suggesting we really need to take a harder look at its role regarding long-term neurodevelopmental outcomes in kids.
And that's our mission today, isn't it? We're bypassing the single studies, going straight to the synthesis level, these umbrella reviews that pull together dozens of primary studies.
Yep.
We want to focus specifically on that link. Maternal prenatal acetaminophen use and the risk of, well, two key outcomes: ADHD, attention deficit hyperactivity disorder, and ASD, autism spectrum disorder.
What is the most rigorous data actually telling us? That's the question.
And just to give a sense of scale here, we're not talking about small numbers. One big review synthesized data covering, what was it, around 220,000 mother-child pairs?
That's right, a huge data set. And the initial signal, well, it's hard to ignore. That review found positive associations between the mom using acetaminophen and potential adverse neurodevelopmental effects,
Including ADHD and ASD.
including ADHD, ASD, even things like lower IQ scores.
Mhm.
In 26 out of the 29 studies they included, that's a really high proportion.
Wow. Okay, 26 out of 29 across that many people. That's definitely not just statistical noise. That demands attention.
Mhm.
Okay, let's unpack this.
Agreed.
Where should we start? Maybe where the evidence seems clearest. The ADHD link.
Yes, I think that makes sense because really the core consensus from these big umbrella reviews is pretty clear on this one. There's a consistent, statistically significant association between prenatal acetaminophen exposure and increased risk of ADHD outcome.
It keeps showing up.
It keeps showing up time and time again across multiple high-quality analyses. It seems quite robust.
So when we say these are high-level reviews, looking at other reviews, they must have some pretty solid numbers, right? How do they actually quantify this risk?
They do, and the numbers are remarkably consistent, actually, for observational research. So let's take one example, an umbrella review by Koch and colleagues back in 2022. They looked at 5 separate meta-analyses on this specific link,
Okay.
and they found a pretty tight range for the risk ratio, the RR. It fell between 1.08 and 1.34.
Right, and just to clarify for everyone listening, an RR of 1.0 means no difference in risk.
Okay.
So 1.34, that's like a 34% increased relative risk compared to no exposure.
Exactly.
Now, some people might hear a 1.34 and think, okay, that's maybe not a huge number, not like doubling or tripling the risk. How do researchers defend calling an RR around, say, 1.25, which seems to be another common finding, robust or convincing?
That's a really important point and a fair question. In some contexts, like a trial for a very rare condition, 1.25 might seem modest.
Right.
But here, we have to consider 2 things. First, the exposure to acetaminophen use in pregnancy is incredibly common. We said 65%, maybe more.
Okay.
Second, the outcome, ADHD, affects millions of children worldwide. It's also very prevalent. So even a seemingly modest increase in relative risk, like 1.25, when applied to millions and millions of exposures.
It translates into a very large number of actual cases, a big public health issue.
Precisely. The absolute risk increase becomes significant due to the sheer scale of exposure. And the confidence in that 1.25 number is boosted by the rigor behind it. Another major umbrella review, this one by Kim and colleagues in 2020, they landed on that specific relative risk of 1.25. But critically, it came with a very, very tight 95% confidence interval, 1.17 to 1.34.
Meaning they're very confident the true risk lies within that narrow window.
Extremely confident. That's statistical precision. The fact that different high-level reviews using slightly different methods are converging on roughly that 25% increased risk, that really strengthens the finding.
It adds a lot of credibility.
It does. And Kim and colleagues actually classified this specific APAF-ADHD association as having convincing evidence, Class I.
Class I, that sounds like the highest level you can get in this kind of research.
It pretty much is for observational epidemiology. To get that Class I rating, the association can't just be statistically significant. It has to hold up under really stringent testing, like sensitivity analyses designed to try and break the finding.
So what kind of stress tests did they put it through?
Well, they did things like restricting the analysis to only the highest quality study designs. For example, just looking at prospective cohort studies.
The ones that enroll moms before they take the drug, not relying on memory years later.
Exactly. Much stronger design. They also looked only at the results that had already been most heavily adjusted for potential confounding factors, things like the mom's education, health conditions, lifestyle.
Trying to rule out other explanations.
Right. And the key thing is, even when they sliced the data these ways, looking only at the best subsets, that 1.25 association, that increased risk for ADHD, it held firm, it didn't disappear.
So it wasn't just an artifact of maybe weaker studies or unmeasured factors?
That's what the evidence suggests. They really tried to poke holes in it, and the signal remains strong.
Okay, that's compelling. But there's more, right? Beyond just yes or no exposure. Something about the amount or duration.
Yes, and this is often considered very powerful evidence in epidemiology, even though it's not definitive proof of causation, a dose-dependent relationship.
Here's where it gets really interesting. Because if taking more of the drug or taking it for longer leads to a higher risk, that makes it harder to blame something else entirely, doesn't it?
It makes it much harder. Why would, say, having a fever for 3 weeks perfectly correlate with taking APAP for 3 weeks? But the APAP association is stronger and graded. It points more towards the drug itself.
So what do they find about the dose or duration?
It seems duration is key. Several studies, including some really well-designed ones like sibling-controlled studies, which help account for genetics and family environment,
Okay.
they found that the risk wasn't uniform. It was significantly higher for children whose mothers used acetaminophen for a longer duration. Often this was defined as using it for more than 28 days total during the pregnancy.
More than 28 days. So we're not just talking about taking it for a headache for 2 days.
Exactly. That longer-term exposure, maybe for chronic pain like back pain or persistent migraines throughout pregnancy, that seemed to be associated with substantially poorer neurodevelopmental outcomes, including ADHD, compared to shorter-term use.
That really shifts the conversation, doesn't it? From managing an occasional fever to potentially managing chronic conditions during pregnancy. The concern level seems to ramp up significantly with that longer duration.
It absolutely does.
Yeah.
It suggests that the clinical focus might need to be different for acute versus chronic use. Finding alternatives for that long-term management becomes perhaps more critical based on this data.
Okay, but we have to tackle the big question head on. Whenever you have studies like this linking a common drug to a health outcome, the immediate critique is confounding by indication.
Always. It's the central challenge.
Is it the acetaminophen causing the ADHD risk? Or is it the reason the mother took the acetaminophen? You know, a serious infection, high fever, chronic pain, inflammation, things that might themselves affect fetal development.
Exactly. Could the APP just be a marker for underlying maternal illness or distress? That's the crux of the issue. A severe illness during pregnancy can impact the developing fetus, absolutely. So researchers have to be incredibly careful to try and separate the effect of the illness from the effect of the treatment.
So that convincing evidence classification we talked about really depends on how well the original studies controlled for these confounders. What kind of factors did they actually account for?
They were remarkably thorough in the primary studies. They adjusted for a whole host of things. Sociodemographics, obviously maternal education, age, income, pre-pregnancy weight or BMI, marital status.
Standard stuff?
Right. And lifestyle factors, smoking, alcohol use during pregnancy, other substance use, very important. And also, crucially, maternal mental health. Things like anxiety, depression, or a history of psychiatric illness, because those can link both to medication use and child outcomes.
Okay, but the absolute key must be adjusting for the reasons for taking APAP, right? The indications themselves.
Precisely. That's non-negotiable.
Yeah.
The studies specifically control for things like maternal fever during pregnancy, reported infections or inflammation, conditions like musculoskeletal pain or joint diseases, and just general pain as an indication.
So they'd statistically try to remove the influence of, say, having a high fever and then see if the APAP link still remained.
That was the goal in all the adjusted analyses, to see if the association between AP-OMP ADHD persisted after accounting for the presence and maybe even severity of the underlying condition being treated.
And we know the association did hold up even with these adjustments. So what was the next level? Was there a more sophisticated way they tried to tackle this potential leftover confounding?
Yes, there was. It's a technique called target adjustment sensitivity analysis. Sometimes it's referred to as Schneeweiss's rule-out approach.
Okay, that sounds complex. Break that down for us.
It's basically a statistical method designed to estimate how strong an unmeasured confounder would have to be to completely explain away the observed association.
So it's like asking, what if there's some hidden factor we didn't measure, like a specific virus or a genetic thing linked to pain and ADHD? How powerful would that hidden factor need to be to make this APAP ADHD link disappear entirely?
Exactly that. You model a hypothetical perfect confounder, one that's associated with both taking APAP and developing ADHD. Then you calculate what its effect size, its own risk ratio, would need to be to make the observed APAP-RR of 1.25 essentially go down to 1.0, no effect.
And what did that calculation show? How strong would this hypothetical confounder need to be?
They found that for residual confounding, say, from unmeasured severity of fever or pain, to fully explain the link between APAP and ADHD, that confounder would need to have an RR of 3.0 or greater.
Wow. So the underlying illness itself would need to triple the risk of ADHD all on its own to make the AT effect vanish statistically.
Correct. And based on what we currently know, most researchers consider it pretty unlikely that common indications like fever or non-chronic pain, even if imperfectly measured, would have such a massive independent effect, an RR of 3.0 or more, on ADHD risk in these populations.
So while you can't rule out some residual confounding, it probably doesn't explain the entire 25% increased risk they're seeing.
That's the argument this sensitivity analysis supports. It suggests the APP itself is likely contributing independently to the risk, not just acting as a marker for the illness.
That's a really clever statistical approach, but, statistics can only go so far. Did any studies manage to get around the whole issue of relying on moms remembering if they took Tylenol months ago?
Yes, and these are particularly compelling. Some studies used objective biomarkers. They actually measured acetaminophen and its metabolites directly in the baby's first stool, the meconium.
Ah, right. Meconium forms over the second and third trimesters, so it gives a biological snapshot of exposure late in pregnancy, completely bypassing maternal memory or reporting bias.
Precisely. It's an objective chemical measure of what the fetus was actually exposed to.
Yeah.
And these studies, the ones using meconium biomarkers,
What did they find?
they also supported the association with an increased risk of ADHD. But even more interestingly, they started to provide a link to the brain. The increased ADHD risk associated with higher meconium APP levels appeared to be mediated by measurable differences in brain connectivity, specifically in the frontoparietal network.
The frontoparietal network. What does that part of the brain do? Why is that significant for ADHD?
That network is essentially the brain's executive control center. It's critical for things like working memory, planning, impulse control, decision-making, and crucially, sustained attention.
So the core functions that are typically impaired in ADHD.
Exactly. Disruptions in the structure or the functional connectivity of that specific network map very closely onto the symptoms of ADHD. So finding that chemically measured fetal exposure to APP is linked to changes in those exact brain circuits, well, that provides a very plausible biological pathway connecting the exposure to the outcome. Now, we focused heavily on ADHD because the evidence there reached that convincing threshold. But we also need to talk about autism spectrum disorder, ASD.
Right. The umbrella reviews often looked at both.
They did. And APAP exposure is frequently identified as a potential robust environmental risk factor for both conditions. This has led some researchers to wonder if there's some kind of shared vulnerability, maybe a transdiagnostic feature that APAP exposure might be triggering.
A common pathway affecting both. But you mentioned earlier the evidence for ASD wasn't quite as solid or consistent as for ADHD. Why the difference?
There are a few reasons. A major one is methodological heterogeneity, basically. The studies looking at ASD were more varied in how they defined and measured it. ASD is inherently a broader spectrum, and it's often diagnosed later than ADHD, sometimes not until age 3 or even later.
So that longer time gap between the prenatal exposure and the diagnosis makes it harder to study.
It does. It introduces more time for other environmental factors to play a role, more potential confounders. And the way ASD was measured in the original studies varied a lot too. Some use simple parent questionnaires or screening tools, others use school records, and some use full clinical diagnoses based on standardized assessments in a hospital setting.
And trying to pool data from such different measurement methods, that creates noise.
Exactly. It makes the combined results, the pooled estimates, and the meta-analyses less cohesive. The statistical signal gets weaker and more variable. So while there's a signal suggesting an association between prenatal APP and ASD risk, it generally doesn't reach the same level of consistency or strength as the ADHD finding because of this measurement variability.
That makes sense. Measurement matters. Okay, let's talk genetics for a moment, because the overlap between ADHD and ASD is kind of fascinating, isn't it?
It really is. Clinically, on the surface, there's often a huge amount of overlap in symptoms. You see kids with challenges in attention, social interaction, maybe repetitive behaviors. The symptoms can really blur the lines between the two diagnoses.
So behaviorally, they look quite similar sometimes. You'd assume they share a lot of the same underlying genetic risks.
That would be the logical assumption based on observation. But then, when you look at the large-scale molecular genetic studies,
Like the PGC, the Psychiatric Genomics Consortium.
exactly these huge international collaborations analyzing DNA from hundreds of thousands of people, they actually find a much weaker genetic correlation between ADHD and ASD common variants than you might expect from the symptom overlap.
Interesting. So their core genetic architectures might be more distinct than they appear clinically.
That seems to be the case, at least for the common genetic variants studied so far. There's some shared genetic risk, definitely, but also quite distinct genetic pathways for each disorder.
So why is that genetic distinction important when we're talking about an environmental exposure like acetaminophen?
It's important because if these two disorders, which seem somewhat genetically distinct, are both associated with the same environmental risk factor, prenatal APIP exposure, it really strengthens the idea that the environmental factor might be hitting some kind of shared biological pathway downstream from the initial genetic risks.
Ah, so the APIP exposure might be that transdiagnostic feature affecting a common biological process relevant to both ADHD and ASD, even if their genetic starting points are different.
That's one leading hypothesis.
Mm.
The environmental factor could be bridging the gap, impacting a process crucial for both types of neurodevelopment.
That really elevates the potential significance of this environmental link.
Okay, beyond ADHD and ASD, did the reviews look at other brain-related outcomes, like intelligence?
Yes, they did. Several studies specifically investigated IQ, and some high-quality ones found a link between prenatal APAP use, particularly exposure in the first or second trimester, and slightly lower performance IQ scores in children later on, maybe around age 5.
Performance IQ specifically, not overall IQ or verbal IQ.
Yeah.
Right. Performance IQ tends to measure things like nonverbal reasoning, visual spatial skills, processing speed, and executive functions planning, organizing, working memory. It's less about vocabulary or stored knowledge.
And that links back to the frontoparietal network again, doesn't it? The executive control center.
It does seem to align. The fact that the association was specifically with performance IQ, and some studies suggested this was independent of whether the mother had a fever, points towards a potential impact on those executive function circuits rather than just a general effect on intelligence.
So again, a more targeted effect seems plausible.
It seems that way. The strongest signals consistently point towards ADHD, potentially ASD, though with those caveats, and these performance IQ executive function measures. When researchers looked at broader outcomes like general measures of communication delay or motor skills delays, the effects tended to be smaller, less consistent, and often hovered right around the no effect line after all the statistical adjustments were made.
So the data really seems to zero in on that attention, executive function, and possibly social communication cluster of skills.
That appears to be where the most robust associations lie, based on the current synthesis of evidence.
Okay, so we've got this compelling statistical association, especially for ADHD. We've addressed the confounding issue with sophisticated methods and even biomarker data pointing to brain changes. Now, we need the why. What's the biological story here? How could a drug like acetaminophen, considered safe for so long, actually disrupt brain development in a way that affects attention and executive function?
Right, the biological plausibility. It really starts with how the drug gets to the fetus in the first place. Acetaminophen has the right chemical properties.
Yep.
It's lipid-soluble, doesn't ionize much, and it's a small molecule.
Meaning it can cross barriers easily.
Exactly. It readily crosses the placenta. It doesn't just trickle across. It gets into the fetal circulation in significant concentrations. So the developing brain, the central nervous system, is directly exposed.
So it's not filtered out effectively. The developing brain gets a real dose. How does the fetus handle it compared to an adult?
And that's where the problems really start. It leads us to what many researchers believe is the core mechanism, oxidative stress, driven by a depletion of glutathione.
Okay, unpack that. Oxidative stress and glutathione.
So when anyone metabolizes acetaminophen, primarily in the liver, A small amount is converted into a toxic byproduct. It's called NAPQI. That's the metabolite that causes liver damage
Right.
in an overdose situation, right?
That's the one. Normally, in an adult with a healthy liver, NAPQI is quickly neutralized by an antioxidant called glutathione. Glutathione grabs onto it, detoxifies it, and allows it to be safely excreted.
Okay, so glutathione is the defense mechanism.
It's the primary defense. But during pregnancy, there's a major issue. Actually, two issues. First, the fetal liver is still developing. It doesn't have the full capacity, the full set of enzymes to process ACase efficiently or to produce enough glutathione itself. So NAPQI can build up more easily in the fetus. And the second issue.
The second issue is that pregnancy itself puts a major strain on the mother's glutathione levels.
Really? Why is that?
Well, pregnancy is just a state of high metabolic demand, high oxidative stress naturally. Building a placenta, supporting fetal growth, uses up resources. Studies have shown that pregnant women can have significantly lower serum glutathione levels compared to non-pregnant women, maybe up to 26% lower on average.
Wow.
And even more striking, some research suggests glutathione levels can drop by as much as 80% between the first and second trimesters.
80%. That's a massive drop right when crucial brain development is happening.
It's a huge deficit. So you have this situation, a developing fetus, highly sensitive brain wiring going on, exposed directly to APAP, which produces this toxin NAPQI. And at the same time, the main defense system against that toxin glutathione is severely depleted in both the mother and likely the fetus.
So even a normal therapeutic dose for the mother could potentially overwhelm the fetus's limited detoxification capacity.
That's the concern. It creates a scenario where the fetus is much more vulnerable to NAPQI-induced oxidative stress.
And what does that oxidative stress do at the cellular level in the brain?
A key target appears to be the mitochondria.
The energy factories of the cell.
Exactly. NAPQI is known to directly damage mitochondria. It can bind to mitochondrial proteins, even mitochondrial DNA, and impair their function. And mitochondria aren't just powerhouses, they're also major sources of reactive oxygen species, or ROS, the molecules that cause oxidative stress when they're out of balance. Damaged mitochondria produce even more ROS, creating a vicious cycle.
And mitochondrial problems, that links back to ADHD and ASD hypotheses, right?
It does. There's a growing body of research suggesting that disruptions in cellular energy metabolism might be fundamental to the origins of both disorders. The brain is incredibly energy hungry. Neurons need constant power for firing, for making connections, for plasticity.
Especially those high-demand networks like the frontoparietal one.
Precisely. If the mitochondria in those critical brain cells are damaged by NAPQI and oxidative stress, they just can't produce energy efficiently.
Right.
This could lead to suboptimal functioning, especially during demanding tasks.
How does that translate into, say, difficulty paying attention? Is there a specific theory, like that lactate shuttle idea?
Yes, the astrocyte neuron lactate shuttle hypothesis is one compelling idea. Neurons rely on support cells called astrocytes to provide them with lactate, a key fuel source, especially during sustained activity.
Like concentrating on something.
Exactly. If APAP-induced mitochondrial damage occurs in either the neurons or the astrocytes, it could impair the production or utilization of this lactate fuel. The neurons essentially run out of gas during tasks that require sustained mental effort.
Leading to fatigue at the neural level, which we then see as difficulty focusing, distractibility, poor impulse control.
That's the proposed link. It connects the molecular damage from APAP exposure directly to the core behavioral symptoms of ADHD. The biological mechanism aligns with the observed outcome.
Okay, that oxidative stress mitochondrial pathway seems central. Were there other potential mechanisms explored?
Yes, definitely. Acetaminophen has also been proposed to act as an endocrine disruptor. It might interfere with steroid hormones that are critical for organizing the developing brain.
Hormonal disruption.
Right. And then there's evidence, mostly from animal models, linking prenatal APAP exposure to lower levels of BDNF, brain-derived neurotrophic factor.
Which is vital for neuron growth and survival, making connections.
Absolutely critical for brain development. Lower BDNF is not good. Animal studies also showed alterations in the dopamine system, changes in dopamine metabolism. And dopamine, of course, is the key neurotransmitter implicated in ADHD.
So multiple potential hits on important brain systems.
Potentially. And finally, there's some suggestion that APA might indirectly affect things by activating the mother's immune system or perhaps the fetal endocannabinoid system. Both of those systems are also increasingly thought to play roles in shaping neurodevelopment and might be involved in the pathways leading to ADHD and ASD.
Wow. Okay, this has been a really deep dive into some complex and honestly quite concerning research. If we try to synthesize this, we've got this strong, convincing statistical link between prenatal acetaminophen use and ADHD risk, backed by serious efforts to rule out confounding.
Right, including those sensitivity analyses and the biomarker studies.
Then we have objective evidence from meconium-linking fetal exposure to changes in that critical frontoparietal brain network involved in attention and control.
A potential physical correlate in the brain.
And finally, we have these biologically plausible mechanisms, primarily centered on that perfect storm of oxidative stress from NAPQI buildup during a period of profound glutathione depletion in pregnancy, leading to mitochondrial damage in the developing brain.
That really summarizes the key pillars of the evidence. It paints a complex and, as you said, ethically challenging picture. The sheer volume and consistency of the data mean we can't simply dismiss it.
But we also have to remember the limitations the researchers themselves point out, right?
Absolutely. We mentioned the variability in how ASD is measured across studies. That's a big one. And also the fact that a lot of these large foundational cohort studies were done primarily in populations of European descent.
So we need more research to know how well these findings apply to other populations globally.
Definitely. Generalizability is always a question that needs further investigation.
So given this strong signal, particularly for ADHD, even with the caveats, what's the takeaway? What's the consensus message coming from the scientific community that's been reviewing all this? Is it, don't take acetaminophen?
And no, it's definitely not that simple, and that's not the message. Because we also know that untreated high fever in the mother is a risk for the fetus. Acetaminophen remains a really important tool for managing fever during pregnancy.
So it's not about banning it.
It's not about abolition. The consensus leans much more towards minimization of exposure. The focus is shifting towards using acetaminophen during pregnancy only when it's truly clinically necessary.
Avoid casual or unnecessary use.
Exactly. And perhaps most critically, based on that dose duration data, avoiding prolonged chronic use whenever possible. If someone needs pain relief for weeks or months, exploring alternatives should be a priority.
So shortest duration possible, lowest effective dose, only when needed.
That's the essence of the precautionary principle being applied here. The evidence is strong enough to warrant this caution, to inform clinical guidelines, and to encourage careful conversations between pregnant individuals and their healthcare providers about the risks and benefits in their specific situation.
So what does this all mean? The deep dive is complete.
And that's a wrap on this episode of MedDecode. I really appreciate you joining me today. If something we talked about sparked your interest, or if there's a medical term that's been bugging you, let me know in the comments. And if you enjoyed this, be a pal and share it with a friend. We'll catch you on the next one.